Hello, everyone. Thank you so much for the invitation from NIH. My name is Dr. Gunnar Gottschalk. It's so wonderful to be here to share some of our recent work and to introduce what our plans are moving forward in the next couple of years. Simmaron Research is a 501c3 nonprofit biomedical research institute. We have and really try to focus on multidisciplinary translational science. We collaborate directly with Dr. Dan Peterson at Sierra Internal Medicine, where for many years we've worked and collected samples and executed clinical trials in Incline Village, Nevada. Our more recent change has been to bring and start the Simmaron Research and Development Laboratory, which is based at the University of Wisconsin, Milwaukee campus. And our primary focus there is to translate some of the findings from the Incline Village and other clinical collaborations into the laboratory. And that includes looking at biomarkers and animal models, as well as drug discovery. A huge benefit of our spot at University of Wisconsin-Milwaukee is we are also a part of the Milwaukee Institute for Drug Design. So that is a group that essentially is a group of chemists that is focused on drug development in multiple different types of disease settings. And so while we're very much new to the UWM system, and certainly chronic fatigue syndrome, ME/CFS is very new to the UWM system, we're extremely excited to be there and leverage opportunities to try to find new mouse models, new cures, et cetera. This is our team. You'll be hearing from Dr. Avik Roy, who's the Chief Scientific Officer following this. Of course, Dr. Daniel Peterson is still the clinical PI at Sierra Internal Medicine. And I am myself, as well as the Executive Director, a Principal Investigator. We have two primary focuses coming up in 2023 that Avik will get into in a moment, looking at mTOR activation and autophagy impairment. We've published a manuscript looking at the target ATG13, which is a very important molecule for the induction of autophagy. Autophagy is a process in the cell that helps remove specific types of proteins from intracellular waste products, and that is one of our projects. And then secondarily, we're trying to translate that finding of autophagy impairment in ME/CFS patients into animal model development, specifically looking at cardinal symptoms of ME/CFS. I want to preface this by saying we're not trying to make a mouse model to understand all of ME/CFS. That would be impossible. Our primary focus is to look at cardinal symptoms, including post-exertional malaise. We feel that in other fields of neuroscience, including Alzheimer's disease and Parkinson's disease, there has been a lot of progress made with respect to drug development and the understanding of illnesses through animal models. And that's what we plan on doing. And specifically because both Avik and myself have a background in Parkinson's disease and Alzheimer's disease, we felt that it would be a wonderful contribution to this field. Like Vicki said, we're very thankful for the R21 funding. We've also had support from the Solve ME Ramsey Award program grant, which is another competitive grant award system. And also through our local connections to the Parasol Tahoe Community Foundation. And finally, a thank you to all of the donors who also support our work. Thank you so much. And with that, I'm going to turn it over to Dr. Avik Roy, and he's going to talk about our current progress. Avik?
On behalf of Simmaron Research, I would like to say thank you to NIH for this opportunity. Today, I'm going to talk about our ongoing research. It's in very pilot stage right now. How mTOR activation and subsequent autophagy impairment are critical in the context of ME/CFS. So let's start with some background. ME/CFS is a chronic multi-system illness characterized by severe muscle fatigue, pain, and brain fog. Cardinal symptoms of this disease are post-exertional malaise, which is a severe fatigue, pain, and dizziness post-treadmill exercise, and orthostatic intolerance, which is characterized by sudden drop of blood pressure while maintaining the upright posture. Despite intense investigation, until now, the molecular mechanism of this disease is poorly understood. However, there are some potential contributing pathways that are believed to be involved in the pathogenesis of ME/CFS. Some of these are mitochondrial toxicity, upregulation of inflammatory molecules, myelin abnormalities, and autophagy impairment. There are challenges that impede the understanding of this disease are lack of sufficient post-mortem biospecimens. This disease is highly heterogeneous in nature in terms of its etiology, and there is no reliable disease model. So our goal is to make a disease model to study some of the pathological features of ME/CFS. In our context, it is post-exertional malaise. So we want to achieve this goal by 3 different strategies. The first one is a drug-induced animal model in which we will administer drug in a mouse to induce PEM. The second is transgenic mouse model where we manipulate a host gene to induce post-exertional malaise. And number 3 is a disease-relevant model where we want to induce a virus or a plasma-infused mouse model. So let's talk about a drug-induced mouse model first, because this is the study we are doing right now. So the targeted pathway is autophagy. So let's give a brief background of autophagy. Autophagy is a cellular quality control process by which the defective mitochondria and biologically inactive proteins are enclosed in a vesicle known as autophagosome and then targeted to lysosome for further hydrolysis. So if you look at the entire process, you can see that there are multiple proteins that are involved in the process in different layers. And many of these proteins are tightly connected in disease and regular health. For an example, like Beclin and LC32, these 2 proteins are reported to be upregulated in the serum of patients with acute ischemic stroke. Beclin and P62, these 2 proteins are upregulated in the serum of patients with pulmonary embolism. ATG5 has been reported to be upregulated in AD patients, Alzheimer patients. Alpha-synuclein is reported to be upregulated in Parkinsonian patients. So many of these proteins are upregulated in serum during different metabolic disorders. However, the role of this pathway, autophagy, and its impairment is not studied well in ME/CFS. So to understand the autophagy impairment in ME/CFS patients, we performed an antibody array. Antibody array is a procedure in which a membrane is engraved with antibodies of different metabolic proteins. So in this, the membrane is engraved with 34 antibodies of 34 different autophagy-related proteins. And after that, that membrane is challenged with the serum of healthy control and patients. So we have 2 different representative images. These are collected from 2 different case control studies. The first one is like an age-matched and gender-matched healthy male and patient. And the second one is age and gender-matched female patients and healthy control. So in both cases, you can see that the myriads of proteins, there are a lot of autophagy-related proteins that are upregulated in patient serum. However, the ATG13, which is shown in this rectangular enclosure, in both cases, consistently upregulated. So we are intrigued about the upregulation of ATG13 in patient serum, in ME/CFS patients. So subsequently, we performed an ELIJA study in 10 different cases, like 10 healthy controls and 10 age-matched patients, and we saw that ATG13 is consistently upregulated in the serum of patients, in the serum of all ME/CFS patients. We further continued to understand the metabolic regulation of ATG13, and we did immunoblot studies where we found that ATG13 is heavily phosphorylated in its serine residue, and that phosphoserum is strongly elevated in patients in ME/CFS serum. So we further continued to show that the ATG13, when it is phosphorylated, it's become inactive, and it goes to the serum of ME/CFS patients. And it also can induce a ROS and nitric oxide production via RAGE-mediated induction of ions. We published this piece of work in Molecular and Cellular Neuroscience last year. But our current study is just after that, it starts just after that. So until now, we know that ATG13 is inactivated, and when it gets inactivated, it becomes phosphorylated, and it is elevated in the serum, and it causes autophagy impairment, and that subsequently causes the other downstream metabolic disorders, such as mitochondrial dysfunction, cell death, and inflammation. But we really don't know what causes ATG13 inactivation. So what is the upstream molecule that causes ATG13 inactivation? So then we look at the signaling pathway, and we found that there is a cytosolic protein. Its name is mammalian target of rapamycin, or mTOR. It's a kinase. It's a serine threonine kinase, and it has a dual role in the cell. So when it gets inactive, then ATG13 can participate with the autophagy, and that is very integral process for a healthy tissue. But when mTOR is active, then it phosphorylates ATG13 in multiple serine residues that causes the inactivation of ATG13, and ATG13 eventually aborts that active complex formation, and that causes autophagy impairment. And we also hypothesize that mTOR, since it's a kinase, it can regulate many downstream pathways that can induce different inflammatory molecules in the muscle tissue. For an example, IL-6 and RANTES, and that might contribute to the pathology as well. And we want to test this hypothesis by this compound, and we put the code name that SIM501. The SIM501 is a potent mTOR activator, and we want to see that what happens if this compound is administered in mouse. So we took 3 different doses, like 2, 5, and 10 milligram per kilo body weight. And we administered this drug in mouse via gavage, via oral route, for 14 days. And after 14 days, we plan to measure muscle fatigue through EMG measurement, post-exertional malaise, pre- and post-treadmill regime. And we also want to see the attention deficit and brain fog in these mice. Interestingly, after 5 consecutive days of feeding, we saw there is severe weakness in young female mice. So these female mice, they show severe fatigue with periodic episodes of freezing. Their movement is slow, and at the end of 14 days of feeding, they show a severe loss of their movement, severe fatigue, and a hypothermia. So we are going to the next slides where we can discuss more about this. So after 14 days of feeding, what we found that the young female mice, they didn't gain body weight, and they have sufficient loss of muscle mass. And when we performed an EMG analysis, we saw that the SIM501 treated mice, they display a certain pattern of muscle wave, which we call the marching soldier muscle wave. And that kind of muscle wave we see when there is inflammatory demyelination in muscle tissue. So muscle tissue are getting innervated by different peripheral nerves, So when there is demyelination in these nerves, then we see this kind of muscle wave. Whereas for the vehicle-fed mice, we see there is like a normal, a regular kind of muscle wave here. And we also measured grip strength. And what we saw that increasing doses of SIM501 caused a severe loss in grip strength. And we also measured overall movement in the mice, and we saw that yes, that SIM-treated mice, they have shown a significant loss of their movement with periodic freezing. Now we wanted to see the molecular evidences, like what exactly is happening in the tissue level. So we performed H&E staining just to see that mononuclear infiltration in the tissue, and we found that in biceps muscle in those mice, there is severe infiltration of mononuclear cells. And when we performed, when we characterized that what kind of T cells or what kind of mononuclear cells are infiltrated, we didn't see neither CD4 nor CD8 T cells were there in the muscle parenchyma. However, we observed that there is a severe infiltration of macrophages in the muscle tissue after SIM treatment. We further characterized those macrophages because there are two different types of macrophages. One type of macrophage is inflammatory and toxic. We call it M1 macrophages. And the other macrophages, they're called M2 macrophages, which are kind of protective, and they're anti-inflammatory. And what we found that those infiltrated macrophages, they have expressed CD40, which is a marker of M1 type of macrophages. So those macrophages, they are inflammatory macrophages. And so we are characterizing more on these cells. And so this is the what actually I can share with you right now. So the next question we asked that whether these mice can show post-exceptional malaise, which is a very key pathological hallmark of ME/CFS. So to do that, we let these mouse run on treadmill. So this is before pre-treadmill activity of these mice for the vehicle and SIM-fed mice, and we can see that vehicle-fed mice, they are totally active and they don't have any kind of movement deficit, whereas SIM-fed mice, they have a partial movement deficit compared to vehicle-fed mice. However, when we make them run in a treadmill for 14 RPM per 15 minutes, immediate after that, both vehicle and SIM-fed mice, they have a transient fatigue that is quite expected. However, these mice, they have more fatigue immediate after the treadmill exercise. And then we gave them 2 days of rest. And after 2 days, we saw that the vehicle-fed mice, they completely regained their movement, whereas these SIM-fed mice, they are still very much inactive and they have still movement deficit. So this is kind of very intriguing and very interesting finding, actually, what we found so far. And we are doing more molecular study on that, on these mouse. So what kind of inflammation is happening, and if there any inflammatory demyelination in the muscle nerves. So all these analyses are going on right now. But what I can say that there are some limitations of this drug-induced model because the fatigue is transient, because those mice, they come back to the normal schedule after 1 month. And the direct role of ATG13 is not clear in this mouse because the drugs seem that can inhibit a different other targets, so that we don't know. So to nullify these drawbacks, we plan to do a ATG13 knockout mouse model, and where we are expecting the fatigue is much more stable, and where we can establish the direct role of ATG13. But on the other hand, that ATG13 whole body knockout mouse, they also show some kind of growth deficit, cardiomyopathy, and less viability. So these might confound the understanding of ATG13's role in ME/CFS. So that's why we just apply, and we got a recent R21 NIH grant from NINDS, where we want to make some kind of tissue-specific knockdown of ATG13 with this system, TRILOX system. So we will do muscle-specific mutation of ATG13, brain-specific, and peripheral nervous system-specific knocking down of ATG13. So what we expect? We expect that ATG13 muscle knockout mouse, they will show some fatigue, maybe some post-exertional malaise. Whereas ATG13 brain knockout mouse, they might show some kind of anxiety and attention deficit, and ATG13 PNS knockout mouse, they might show some sort of dysautonomia. So that is our expectation, but time will say that like what will happen in next few months and few years. And so thank you all for your attention. And this is our core team of our animal study. Our executive director, Dr. Gunnar Gottschalk, and this is Dr. Leggy Arnold from University of Wisconsin, Milwaukee. Linda is kind of like, she's our attending veterinarian who is helping tirelessly in our animal studies. And our collaborators are University of Wisconsin, Milwaukee, and Copa Laboratories. And our current study is being funded by SolveME and NINDS. So thank you all for your attention. Thank you.